Okay, so by and large, some drugs are biotransformed largely or exclusively by one specific CYP enzyme. All right. So with, like phenytoin and warfarin, they undergo major hydroxylation by CYP2C9, halothane, and I know we haven't talked about some of these drugs. That's fine. Halothane, which is an anesthetic, is oxidatively debrominated by CYP2E1. Now, you don't need to know that specifically, but you do have to understand the concept. So there's no learning objective on this slide, so don't start memorizing this. You'll go crazy. Um, Other drugs can be biotransformed by several CYPs. Okay, and so there's major metabolic pathways here in terms of so acetaminophen, for example, undergoes dehydrogenation by CYP2E1, but also has activity with CYP1A, 1A2 specifically, and CYP3A4. Likewise, diazepam and dextromethorphan have affinity with other CYP enzymes. All right. So what does this mean clinically? So take a step back. A drug that's biotransformed by multiple CYP enzymes is usually less susceptible to pharmacokinetic interactions by CYP inhibitors, and which often render one specific CYP enzyme in terms of its activity. So therefore, CYPs that remain uninhibited can still eliminate the drug. So all this really says is that, look, if you've got one drug being biotransformed by 5 different CYP enzymes, okay, the likelihood that there's a major drug-drug interaction typically gets diminished, okay? We really see major drug interactions when we see 3, 4, or 5 different drugs all being biotransformed by the same enzyme, and that'd be CYP3A4. Okay, so CYP3A4 biotransforms, as you can see, more than half, probably about 2/3 of the total number of drugs in the hospital. All right, and so we'll spend some time on CYP3A4. Likewise, CYP2D6 also accounts for a large percentage, up to, what is that, 25 or 27% of the drugs that are available. Okay, so these two CYPs are really important, and of course, these are the two CYPs we're going to talk about for this particular lecture. Understand or recall that these metabolic sites are basically within the hepatocyte. Okay, and you can see within the endoplasmic reticular folds are these oxidoreductive CYP complexes, which are listed here and then kind of blown up this way. And so they basically exist with this complex where you can see the CYP enzyme and you can see some energy transfers, and this one's showing NADPH, as well as iron and sulfur molecules being important in some of these reactions. And so, these are coupled to this kind of larger complex within the plasma membrane, and of course, all this is occurring within the hepatocyte. All right. And so, what do we talk about when we say, induction of CYP enzymes? And so, what we're talking about is really we're increasing the synthesis of the enzyme. Okay, so this is at a genomic level, and you can see an increased gene transcription through several different receptors, all of which are listed there. And of course, this has clinical consequences. And so, if you increase the synthesis of the enzyme, several things can happen depending on the drug that's biotransformed by that enzyme, or the drug that's a substrate of that enzyme, said in another way. Okay, so what are these consequences? Well, you can see increased inactivation of the drug after repeated administration. So drug goes in, more enzyme exists to biotransform it, and so the drug becomes more and more inactivated more quickly because there's more enzyme. That's one example. You can see enhanced inactivation of co-administered drugs after repeated administration of the inducer. Okay, and so here we're looking at two drugs being given together. One is a substrate, and one is an inducer. And what we're worried about is what's going to happen to the substrate. Okay, because usually it's the substrate that has the enhanced inactivation. Okay, and in some cases you can see an enhanced toxification of the substrate, which would be the co-administered drug, after repeated administration of the inducer. Okay, so what does this look like? Let's give you an example. So here's your glucocorticoids, or drug A. Understand that the glucocorticoids modify many targets at the genomic level, and interestingly, one of the targets that they do modify is the CYP3A4 enzyme. Okay, they induce it. Okay, and so now the CYP3A4 enzyme is induced. Okay, so there's more of it. And now here's your co-administered drug. So here's your drug B. Okay, so any of these could be drug B. Okay, even verapamil, don't worry about that. So any of these drugs could be drug B. And now you've got more of this CYP3A4 enzyme converting drug B into this modified version of drug B. Okay. Now in some cases this could be good, in some cases this could be bad. It depends what this reaction is. Okay. And in some cases you can see an increase in therapy, in some cases you can see a reduction in therapy. You know, it depends on this particular chemical reaction, and if these drugs are prodrugs, or if this is a toxic metabolite. It completely depends. So it's really hard to predict what the physiologic outcome is going to be when you say drug A and drug B co-administered, where one is a inducer, like the glucocorticoid, and one is a substrate, and the enzyme system is induced. It's very hard to predict that. It's you know, and so the reason that I've gone through all of these different scenarios here, because all of these are possible, and this isn't even a complete list. These are just kind of the three main examples. Okay. So let's talk a little bit about the inhibition of the CYP enzyme. So what are the consequences to CYP inhibition? Well, a little bit longer list. We can see decreased inactivation of the drug, and this can result in increased therapy. So with warfarin, you'd see more bleeding. With a anti-diabetic medication, you can see hypoglycemia. Right? You can see decreased inactivation of the active drug, which will produce toxic or adverse effects. All right? Reduced activation of a prodrug, meaning a lowered therapeutic effect. So it's like they're not even taking their medicine. And you can see a decreased toxification of a drug leading to a reduced toxic effect. And so this would be a drug where you could, usually at this dose, it's toxic, but because of this inhibition, it's actually less safe. It's actually more safe, excuse me. So you can actually see the therapeutic window being increased. Which can happen, not all the time, but it can happen. All right, so that's a good introduction to these two CYP enzymes, CYP2D6 and CYP3A4. Let's end there, and we'll start talking about these enzymes in the next video.